Hi, it's Mrs. Vance again. This is part 3 of Unit 7 notes. This is about the cell cycle, which is part of the bigger topic of cell division. So we have a lot of vocabulary words here, and some of them are going to be easy to confuse with each other. So pay close attention as we go. You might want to stop for a minute and jot some of these words down, but you'll see them as we go through the notes. Just pay close attention. There are lots of vocabulary helps on the in the Schoology unit folder, so be sure and look at the Quizlet in the quizzes there to help you with some of the terms. This is a beautiful picture of two cells that are just finishing the process of cell division, and you can see they look pretty much the same, and that's the whole idea behind cell division. You want the daughter cells, the products of cell division, to be exactly the same, have the same genes, the same DNA, and be able to do the same kind of job, because that's the purpose of cell division, is to provide more cells to replace, either for growth or to replace broken cells or damaged cells or something like that. And so it's important for all cells to be identical as they're being produced, so that you can replace those cells effectively. So this it plays a lot of important roles, like I said, reproduction, in the case of single-celled organisms like bacteria and other single-celled things. For growth of a multicellular organism, you have experienced quite a bit of growth in your life in the past couple of years, and even more so since you were conceived back about 14, 15, 16 years ago from a fertilized egg into a young adult. And then also when you have an injury, then you need to repair and replace cells when you have something like that. So it's really important, and cell division is going on in your body all the time. You're just not aware of it most of the time. Prokaryotes are very simple. Remember, they're just basically some cytoplasm inside a cell membrane and a cell wall, and some DNA with some ribosomes and enzyme processes and things like that, and they're very, very simple. And so they actually reproduce in a very simple way too. The process is called binary fission, which just means dividing in half. And so the big thing to realize about prokaryotes is, number one, they've only got one chromosome. It's a single DNA molecule that's actually in a circle rather than as a line like you normally think about it. And there's some associated proteins along with it as well. But it's also a lot smaller than the chromosomes you find in eukaryotes. So cell division is a much more simple process in bacteria than it is in eukaryotes like us. There are 3 stages of binary fission. First of all, the chromosome has to be duplicated, like we talked about in class. You've got to make sure that each cell has its own full copy of the DNA and that it's a correct copy. Then you need to separate those copies from each other. The cell needs to continue growing and move those copies apart from each other, and then divide into 2 daughter cells that are more or less equal in size and complements, and definitely equal in the case of the DNA. So here we have our bacterial cell, okay, very simple cell again, plasma membrane, cell wall, and there's a chromosome. Duplication can occur there, so you see that here you've got the DNA that's been duplicated, and it looks kind of confusing there, but it's not as confusing as it would be in a eukaryote because you've got a lot less DNA. But each one of these copies basically attaches to the cell membrane on the inside, and as the cell continues to grow, that pulls them apart from each other, and then eventually the cell divides into 2 separate cells, and you end up with 2 daughter cells that are identical identical in the content of their DNA and pretty much identical in size. Very, very simple process, really, really simple. But eukaryotes are a lot more complicated, and so we're going to talk more about eukaryotes, and that involves something called the cell cycle, and then a process called mitosis. You may have heard about mitosis before, maybe seen a little bit about it when you were in middle school, learning a little bit about cells. And so we're going to go into a little bit more detail this year to learn more about how the cell goes about dividing. So the big thing to realize about eukaryote cells is that we have a whole lot more chromosomes and a whole lot more DNA. The cells are much larger and they're much more complex. You've got all those organelles and various things like that. There are a whole lot more genes present in the DNA of eukaryotes. And most of the genes are stored on multiple chromosomes. And so you have to have a way not only to replicate the DNA, but you've got multiple copies of DNA that need to be replicated. and they need to be replicated in a certain way to make sure that each one gets the complete set of the genes that are necessary to run the mechanisms of that organism. Eukaryotic chromosomes are made of chromatin. Chromatin is DNA, basically, that has some proteins that help maintain the structure of the chromosome and help control the activity of the genes. There are several different proteins that are involved in this. It's more detail than we really need to know. We'll talk about a little bit of it, but not a whole lot. But one thing that you need to realize is that most of the life of the cell, when you look at a cell, you can't see the nucleus. You can see the nucleus, you can't see the chromosomes, though, because the chromosomes are kind of relatively unwound because the cell is at work and constantly needing copies of the various genes, the various recipes for proteins that are found in the DNA. In order to get ready for a cell division, the chromatin has to become very compact, and at that point it becomes visible with a microscope, and that's when you see the characteristic chromosome shapes that you're used to seeing in pictures. Here is a cell that is in the process of getting ready to divide. This is an early stage of cell division. You can see definitely the yarn-like structures of the chromosomes as the DNA is becoming more compact rather than diffuse or spread out in the nucleus. So here we see a picture of the chromosomes. This is one chromosome that's composed. This one's already been copied, so it's made of two parts that are identical to each other. These are called the sister chromatids. Replication has occurred. So we're starting off with one chromosome there, and each chromosome sort of in the middle, sometimes closer to the end, has an area called the centromere, which is basically the place where the copied chromosomes are going to be attached to each other. And so here we have our chromosome that's composed of DNA molecules and associated protein molecules that help organize things. When the chromosomes are duplicated through DNA replication, you end up with a chromosome. It's still called one chromosome, but at this point it's made of two sister chromatids. They're identical twins, okay? And then when cell division occurs, then those chromatids separate from each other and become separate chromosomes, each of which goes to one of the two daughter cells from the whole process. So that's the basics of how it works. Again, it's a little bit more complicated than that, but we'll talk a little bit more about the complication as we go on. Before the eukaryotic cell starts to divide, it has to copy all the chromosomes. And so that is where you get the two copies that we call sister chromatids, and that are joined together at a narrow area called the centromere. And when the cell divides, the chromatids are going to separate from each other, and now they'll be full chromosomes, and then be sorted into separate daughter cells. And again, the goal is to make sure that each daughter cell gets a correct copy of each of the chromosomes so that each cell has all the information it needs to be functional. The cell cycle is a sequence of events that goes from the time a cell is first formed from a dividing parent cell until it goes through its own cell division. The length of time for the cell cycle varies depending on what kind of cell it is, what its job is, and so forth. It can be anywhere from, let's say, somewhere in the range of 6 to 8 hours, all the way to years in some cases for some cells. It just depends. There are two main stages in the cell cycle. The first one is called interphase. In interphase, that's actually the vast majority of the time of the cell's life cycle, okay? And that's when a lot of different things happen. There are three sections or three stages of interphase. First one is called G1, and that is the initial growth phase. This is when the cell is growing. It's right after it divides. It's growing and increasing in cytoplasm, doing its normal everyday job. When the cell reaches a certain size or certain kind of conditions, then a signal will send it into the S phase. In the S phase, S stands for synthesis, and that is when the chromosomes are duplicated. That's the process of replication like we discussed in class. And then once the duplication of chromosomes is finished and the DNA has been proofread to make sure that everything is copied properly, then the cell enters what's called the G2 phase. And in this phase, again, the cell continues to grow, but it's also doing all of the things necessary to get ready for cell division. Make sure there's plenty of ATP, because this is a very energy costly process. making sure that all the enzymes and proteins and other things that are necessary for cell division to occur, duplication of some of the organelles and things like that. And when everything is ready to go, then it goes into the last phase of the cell cycle, which is the mitotic phase, and that's where division occurs. In the mitotic phase, you've got mitosis, which is the division of the nucleus, and there are several stages in that we're going to focus on in class. And then cytokinesis, which is division of the cytoplasm and the separation of the two daughter cells from each other. So this picture shows you kind of the way we envision the life of the cell. Right after cytokinesis occurs, here's our brand new cell. One cell in this cell cycle, and this other daughter cell is going to go to its own cell cycle that is similar to this one. A good portion of life here is spent in the G1, also called the first gap stage. And again, the cell is growing, it's increasing in size, it's increasing its cytoplasm, it's doing its job, whatever that job happens to be in the organism. And then once it reaches a certain point, again, it has to do with size, it has to do with space, it has to do with what the needs of the organism happen to be at that time. There will be a signal that will send the cell into the S phase. And during the S phase, that's when DNA synthesis is occurring. That's when the replication of the DNA is occurring. Notice that there's a really large part of time here. It's at least equal to, sometimes not quite equal to, depending on the cell, again, the G1 phase. And then after that is completed and everything is ready to go from the DNA standpoint, then the cell enters the second gap, or the G2 phase, where it continues preparing for cell division. And at the very last, when everything is ready to go, then it enters the mitotic phase, which includes mitosis, division of the cytoplasm, division of the nucleus, and then cytokinesis, dividing the cytoplasm and separating the cells from each other.